I'd like to continue our discussion of chemistry today, moving from kind of the basic chemistry we talked about last time to actually the chemistry of living things, which is biochemistry. So biochemistry is the study of the atoms and molecules of living things, as well as their interactions and chemical reactions. So when we talk about when we talk about biochemistry, we're talking mostly about organic molecules. And organic molecules we're going to define as either being composed of molecules that have a carbon chain, more than one carbon bonded to one another, or being composed of a combination of carbon and hydrogen. And then inorganic molecules would be anything else, anything that doesn't meet those conditions. So if we look at a series of molecules and compounds here and try to figure out if they're organic or inorganic, first looking at sodium chloride, NaCl, we don't see any carbon there, so that would be an inorganic molecule. Glucose, C6H12O6, we see that there are 6 carbons. That is a carbon chain. We see the carbon-hydrogen combination. That's organic. Carbon dioxide, carbon and oxygen. We see 1 carbon but no carbon-hydrogen combination, so that would be inorganic. Oxygen gas is also inorganic, no carbon present. Carbon monoxide, we can see a single carbon but no carbon-hydrogen combination, so that is inorganic. Ammonia, also no carbon, so inorganic. And then methane, CH4, single carbon but a carbon-hydrogen combination, so we consider that an organic molecule. Now, organic molecules, in addition to their chemical formulas, the number of carbons, hydrogens, and so on, that's important, but organic molecules also have structural formulas, so they have complex shapes, and we'll be interested in that as we move forward. So in many organic molecules, the shape of the molecule determines its function. So again, many of these organic molecules or biochemicals are quite complex, and for the most part we'll see a theme in that there's some kind of a basic unit for the molecule, and then those are bonded together. So the initiating unit or simplest unit of a certain type of organic molecule we call a monomer, a single unit or single thing. And monomers can be combined to form dimers, which would be 2 units together, and then dimers can be extended to form polymers, and the prefix poly means many, so this would be many units bonded together. And so as we look at these polymers, then there are 2 general categories of polymers: homopolymers, which are polymers composed of exactly the same unit repeating over and over, and heteropolymers, which are composed of different but similar units bonded together and producing long chains. So just to illustrate this, just using some Legos as an example, let's say that we have a bunch of these blue Legos here, and so those will represent monomer units. And so all of these units are the same, and if we were to bond these together chemically in some way, that will produce a polymer, and we'll learn how they're bonded together here in a couple of minutes. But since the units of which it's composed are identical, we would refer to that as a homopolymer. If we ended up making a molecule in which is composed of different kinds of units, that would be a heteropolymer. Okay, so in taking monomer units and putting them together to form polymers, there's a type of reaction known as a dehydration reaction or condensation reaction that is responsible for this. And so in a dehydration reaction, what happens is that if we have an oxygen and a hydrogen that are adjacent to one another, so as you can see here we have a monomer with a hydrogen and an OH group adjacent to one another, those will bond to form water. And so if we go ahead and move off here a little bit, water will spontaneously form, and then that will leave the 2 units to bond to one another. So this would form a dimer, and then from that dimer you could continue to form longer units, which would be polymers. So again, it's these dehydration reactions where water, again, is essentially dehydrated from the molecules and enters the surrounding solution that forms these dehydration reactions. Now, a hydrolysis reaction is just the opposite of that. If you have a polymer and you want to split it apart, that's what's called a hydrolysis reaction. So we're going to split a water molecule and wedge that into the bond. So again, if we look at this bond up here, what's going to happen is that water molecules will be wedged in there, and then that splits the 2 units apart, and we get back to our monomer units. So when we look at the relationship in life then, we basically ingest polymers, we digest them to monomers, absorb those and then put them to get back together to make human polymers of various types inside of our body. Now I want to show you a quick animation showing you this dehydration synthesis and hydrolysis reactions, and these are mediated by something called enzymes, and we'll talk more about those later. But if we look at this animation, then an enzyme comes in, it's going to bond the monomers, dehydration synthesis puts them together, and so this is how polymers are formed. And then when you eat your food, there are catabolic enzymes that will carry out hydrolysis reactions and split the monomers, or excuse me, split the polymer apart back into the monomers. All right, so looking at specific types of organic molecules. One type of organic molecule is a carbohydrate. And carbohydrates, in the word, if you look at the prefix carbo, that's a reference to carbon. Hydrate is a reference to water. And so when we look at carbohydrates, they generally have a ratio of carbon, hydrogen, and oxygen that's approximately 1 to 2 to 1. Sometimes it's exactly that, but usually it will be close to that. And an example is glucose, C6H12O6. So when we look at carbohydrates and their functions, carbohydrates are very, very important sources of energy, and we'll learn more about that later in the semester. Some carbohydrates function structurally. For example, the cell wall of plants is composed of carbohydrates, a carbohydrate called cellulose. They can also play a role in immune recognition in a variety of different animals. And there are some other functions as well, but those are the major functions of carbohydrates. So again, as we look at carbohydrates in terms of structure, the basic unit of a carbohydrate we call a monosaccharide, a simple sugar, single sugar. And an example would be the sugar glucose. And when we look at this, and so as we look at this example again, so if we look at glucose, again, you have the formula, C6H12O6, that's the chemical formula, but we also have a structural formula. And this is what we refer to as a shorthand diagram. And so as you look at the chemical formula C6H12O6, and you look at this shorthand structural formula showing the three-dimensional shape in 2 dimensions, you're wondering where all the carbons are. It's supposed to be 6 carbons. Where are they? Well, as it turns out, in these kind of drawings of organic molecules, the lines represent covalent bonds. So any place where 2 or more lines come together, so for example here and here and here, that's where carbon molecules would be found. So I'm going to go ahead and plug in those carbons right now. So that's where the carbons would go. So there are 6 carbons if you count this carbon up here as well. And then at the end of each of these lines that are dangling, that's where an OH group would go. We call those alcohol groups. And so we'll have an alcohol group here, 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 and here. And then carbon forms 4 covalent bonds. And hydrogen is always a part of organic molecules. And so there's so many hydrogens bonded to carbon that we normally don't even draw them in. So if we were writing the complete structural formula here, again, the hydrogens would go wherever there is a 4th bond needed to that carbon. So again, in this example, we did have 1, 2, 3 bonds. We needed to add a 4th. So here we've got 1, 2, 3. We need to add in a 4th. Here we've got 1, 2, 3. We need to add in a 4th. Same thing here. Oh, already got that one in. Same thing there. Same thing there. So again, what you normally see are these shorthand notations, but this would be a complete structural formula. And we basically use the same rules over and over and over again. So that would be the actual structural formula for glucose. Now another monosaccharide would be fructose. Now fructose has exactly the same chemical formula, C6H12O6, but it has a different structural formula, it has a different physical shape to it. So fructose is a distinct and different carbohydrate. Galactose would be another monosaccharide that also has the same chemical formula, C6H12O6, but again, a distinct structural formula, similar to glucose, but not identical to glucose. And when we have the same chemical formula but different structural formulas, we call those types of molecules isomers. And again, this is pretty common in organic chemistry or biochemistry. Now, a couple of other monosaccharides, one would be ribose and the other deoxyribose, and they differ only by an oxygen, and these are structural monosaccharides. These are used in the structure of DNA and RNA, which are the composed genetic material. Now, additionally, you can take monosaccharides, put them together to form disaccharides. Some examples of disaccharides would be maltose. And maltose, like glucose, is among the most abundant sugars in nature. So glucose you find in virtually every animal, plant, organism has glucose. And so maltose is also very common. Sucrose is a combination of a glucose and a fructose. Lactose would be a combination of a glucose and a galactose. And so people who are lactose intolerant have difficulty breaking apart that glucose and galactose, those two sugars, from one another. And since you can't break them apart, bacteria will do that for you, and then that produces gas and discomfort and so on. Okay, polysaccharides then would be composed of many sugars, and examples would be starch. And so starch is found primarily in plants. That's how they store their glucose. In animals, multiple repeating units of glucose form something called glycogen. So we store glycogen in our liver and our muscles. And then again in plants also produce a polysaccharide, cellulose. Now what's interesting about starch, glycogen, and cellulose is that they are all three composed of 100% glucose, repeating glucose units. But again, starch is a little bit different from glycogen, and cellulose the arrangement is different still. So as we look at starch, you can see that it's kind of a branching polymer. Glycogen has more branches to it. So again, plants store starch, animals store glycogen. And then again, the arrangement of sugars in cellulose is kind of interesting because animals cannot digest cellulose. We can break apart the glucose molecules of starch, but we can't break apart the glucose molecules of cellulose. And it basically has to do with the arrangement of the glucose molecules. They're arranged kind of upside down to one another, and we lack the molecules needed to separate those apart. So that's something that we'll come back to later, because some animals do utilize cellulose as an energy source, but if they can't digest the cellulose, who does it? Well, they have microorganisms in their gut that do it for them. Now, proteins would be another major class of organic molecule or biochemical. They're composed of repeating units known as amino acids. But proteins are then heteropolymers because there are 20 different amino acids encoded by our genetic material. And proteins have a variety of functions, and their functions are generally based on the physical structure of the protein. So this is one area where structure directly relates to function when it comes to proteins. So the amino acid sequence. So again, at each stage in a protein, you might have a protein that's hundreds of amino acids in length. And you could have one of 20 different amino acids at each position in that protein. So the amino acid sequence of a protein determines the protein shape. That determines the protein function. So that's an important concept, and one that we'll come back to, because this also has evolutionary implications because of the many functions that proteins have. So first, looking at an amino acid, so we can get some idea of how this amino acid sequence affects protein shape. An amino acid, as you can see, takes its name from the fact that it has an NH2 group called an amino group, the COOH group, which is a carboxyl group, which is an acid group. It forms an acid. You'll see there's a central carbon then that has a hydrogen, and then a fourth bond that just says R. And so the importance there is that all of the different amino acids, there are 20 different amino acids, they're all going to differ at this one spot. The 20 amino acids are the same at the other three. They just have 20 different possibilities at this R group, what we call a radical group. So again, it's the amino acid sequence, determines protein shape, determines protein function. That's something we'll see. So when we talk about protein structure, when we talk about the primary structure of a protein, that doesn't refer to a physical structure. That just means the amino acid sequence. So primary structure and amino acid sequence are synonymous. And again, depending on what series of amino acids you have, so if we had methane followed by leucine, isoleucine, proline, alanine, glycine, and so on, that will eventually determine the three-dimensional shape of the protein, because depending what R groups we have, different R groups can interact with one another. They can form ionic bonds with one another. They can form covalent bonds with one another. There may be polar and nonpolar interactions going on. But depending on the types of R groups you have stacked up, they begin to interact with one another. And then that takes the protein into some kind of a three-dimensional shape. So when we look at factors in that affect protein shape, certainly amino acid sequence is part of that. Also temperature. Temperature can affect these side chains, these R groups, and it can affect the types of bonds that form between them. And particularly heat. So heat will change the shape of a protein, and if enough heat is added, it will permanently alter the shape of a protein. And when that happens, we call that denaturing a protein. So and again, it's a permanent change. So for example, when you look at an egg, when you put an egg in a frying pan, initially the egg white is clear, and it's runny. Well, you put it on the fire, you put it in the frying pan, heat it up, it turns opaque, it turns white, and it becomes more rigid. Well, if you take it off the fire, so that's denaturing the proteins, because the egg white is virtually 100% protein. But if you take it off the fire, it doesn't go back to being a raw egg, right? So once you've denatured those proteins, they're done. And so again, that does have health implications as well, because if animals get too hot, if their environment gets too hot, basically they cook. Same thing happens when you get sick. If you have a temperature, if your temperature gets too high, what we're initially trying to do is cook the organisms that are attacking us. We're trying to cook the bacteria or destroy the viruses that are attacking us. But if our temperature spikes too much, we cook ourselves. That's not a good thing. Also, acidity and basicity of a solution can affect the shape of a protein, and so we try to regulate the pH in our bloodstreams. And then also, ultimately, what determines the shape of a protein is our genetic material, DNA nucleotide sequences, but we'll talk about that more later. So as we look at what proteins do then, again, there are some important functions of proteins. I would say the really major important function of proteins is that many proteins functions as enzymes. So we've talked a little bit about chemical reactions, and what enzymes do is that they are what we call protein catalysts. They speed up chemical reactions without being consumed in the reaction, and enzymes are very specific. So enzymes are so important because life would not be possible without enzymes. The chemical reactions that keep us alive are enzyme mediated, controlled by enzymes. They speed up these reactions so they occur at a speed that's comparable with life. And so if those enzymes aren't there, or if they're denatured or destroyed, then we die. Okay, so that's bottom line. Life is not possible without enzymes. So enzymes do basically three things. They're catabolic, in other words, we can take big molecules, make them into small molecules. They're anabolic, take small molecules, make larger molecules, or they rearrange molecules. And just again to give you a quick look at this, using some Lego pieces. So again, a catabolic enzyme would bind a large molecule and break it into smaller molecules. An anabolic enzyme would take smaller molecules and assemble it into a larger molecule. And then some enzymes may just take your molecule and just rearrange it into a different shape. There are certainly other functions of enzymes as well, but that's a pretty simple summary of what enzymes do. Now, the way that enzymes function is that enzymes will have a particular shape to them. And they have what we call active sites that are complementary in shape to some substrate. And so they will bind that substrate, forming an enzyme substrate complex. That will cause a change in the bonding within the enzyme. Typically, the enzyme will actually change shape, put stress on bonds, and in this case, it would break the bond and break our dimer into monomer units here. So you'll notice that the enzyme then goes back to its original shape. It can bind more substrate, carry out the reaction, release the product, and then go back to its original shape. So enzymes can be reused over and over and over again, and this is an important property of enzymes. And as long as there's substrate available to bind, it will generate product. So I actually want to go back to this animation that we saw before. And if we look at our two monomer units, and if we look at an enzyme that's going to put these together, you'll notice that the enzyme bonds the two substrates. In this case, a dehydration synthesis has occurred. Let me show you that again. So the enzyme comes in, bonds, and then it can bond again. But notice that the enzyme shape will not change here. And again, we would have, in this case, a hydrolytic enzyme or digestive enzyme that will do the same thing. But what's important again is that it will have active sites that are complementary to the shape of the substrate molecules that it binds. And so again, the enzyme can be used repeatedly. So if the protein, or excuse me, if the enzyme shape has changed, then the enzyme function is destroyed. So other functions of proteins, some proteins act as transport molecules. They help move molecules across plasma membranes. Some are what we call receptor proteins. They will bind particles that are passing by and then maybe move them into the cell. Some of our proteins are used as hormones in communication. Some of them are used as neurotransmitters in communication between nerve cells. Some proteins are connective. Collagen is one of the most abundant proteins in our body, in animals in general. It connects cells together, bones together, muscles to bone, cells to cells, tissues to tissues. So these connective proteins are really important. This is showing an illustration at the cellular level of some of these types of proteins. And then our immune system, the antibodies of our immune system are composed of proteins. And then also proteins help regulate body fluids. They help control and keep fluid within our bloodstream and help return fluid to our bloodstream and so on. And other functions as well. So again, proteins have a lot of important functions, but all of those functions are dependent upon the shape of the protein. Now, another major group, a third major class of organic molecules then would be lipids. And we also call lipids hydrocarbons. So lipids are composed primarily of carbon and hydrogen. The term lipid means greasy or oily in texture. So most of them are greasy or oily in texture. And the carbon-hydrogen combination, and again, hydrocarbons are composed primarily of hydrogen and carbon. That's a nonpolar combination. And so the nonpolar combination is one that does not dissolve well in water. So oils and water separate as a result of that. Okay, so as we look at lipid functions, again, there are many functions. Again, lipids are important as energy sources, and this will become important when we talk about the Yellowstone ecosystem, because animals, especially as they're preparing to get ready for winter, they want high energy foods, and foods that are high in lipids are high energy foods. Some lipids are structural. Phospholipids form the plasma membranes, for example. Some lipids are important in communication. They can, steroid hormones, for example, are lipids. And then also they play a role in immunity, as well as other functions, but those would be major functions of lipids. So as we look at types of lipids here, fatty acid would be one group of lipids. And fatty acids are long chains of carbon and hydrogen. And these can be 20, 30 carbons in length, more than that even, with an acid group at the end. The COOH is an acid group. So these are known as fatty acids. And so some of these fatty acids we describe as being saturated because there are only single bonds between the carbons, and so the extra bonds are saturated with hydrogens. Some fatty acids are unsaturated. Unsaturated fatty acids have at least one double bond between the carbons, and so we have slightly fewer hydrogens as a result. And both saturated and unsaturated are common in nature. And we also, which would not be very common in nature, but we also, on your food packaging, you may see reference to trans fatty acids. And these are so-called partially hydrogenated fatty acids. And so this is normally the result of manufacturing process. And what it does is help extend the lifespan of fatty acids. So we end up with a slightly different shape in these unsaturated fatty acids. The normal configuration is a cis configuration. This process produces trans fatty acids, and trans fatty acids just aren't as healthy for you, quite honestly. It increases bad cholesterol in your body. They don't have as many biological uses as the cis fatty acids, and so it's a good thing to try to avoid trans fatty acids in your diet. Now, another type of lipid, and again, this is kind of showing us an example of saturated and unsaturated fatty acids in a polymer. But another example of a lipid are triglycerides, also called fats. And so the triglycerides, what the name is trying to tell us is that they form from 3 different fatty acids that are bound to a molecule called a glycerol. So 3 fatty acids and a glycerol form a triglyceride. And these triglycerides then can be formed from either saturated or unsaturated fatty acids. Other types of lipids, phospholipids. Phospholipids have one end that is water soluble, and then the fatty acid tails, which are not water soluble. So fatty acids, when you put them in water, tend to form a bilipid layer, 2 layers of of lipids, and these are an important component of plasma membranes in living cells. And this shows us an example of that bilipid layer. So again, all of our plasma membranes throughout nature are normally formed from two layers of phospholipids with the so-called hydrophobic or water-soluble heads towards the water, and then the fatty acid tails pointed towards one another. So this creates a fairly efficient barrier that prevents particles from moving across the membrane. And these will, again, spontaneously form. Cholesterol would be another example of a type of lipid. So our steroid hormones form from cholesterol, among other things. Cholesterol is also part of plasma membranes. Steroid hormones, as I just mentioned, derive from cholesterol. Waxes, and this is an example of a steroid hormone, what they look like. So again, you can see organic molecules have some complex shapes. Waxes would be another example. Waxes, again, tend to be long chains of fatty acids, and, long chains of carbon and hydrogen. And plants, for example, secrete waxes to keep from losing water. We produce, waxes in our ear and so on to keep insects from crawling into our ears. Prostaglandins would be another category of lipids which are mainly used in regulation. Body temperature, for example, in animals is regulated by means of prostaglandins. And again, there are a variety of other lipids. So again, some of these have energetic functions, some of these have structural functions, but a pretty diverse group. And then lastly, I just want to briefly mention nucleotides. So nucleotides form our nucleic acids. DNA is a primary component of our chromosomes, and RNA helps get the information from chromosomes to actually the production of proteins. So again, we're going to come back to the structure of this later, but again, as we look at a chromosome, for example, the chromosomal structure is formed from DNA. DNA is composed of these repeating units that are called nucleotides. And so again, as we look at the relationship then, it's actually this nucleotide sequence of the DNA chromosomes that determine the amino acid sequence of the proteins, which determines protein shape, which determines protein function. And then following that up even, changes in the DNA then leads to changes in the protein function, and nature decides the value of that, which is one of the driving forces of evolution. So that's it for today. I'll see you next time.